Hey everyone, welcome back. I am absolutely thrilled you're joining me for this explainer today because we are diving into something truly mind-boggling. I want you to just stop and think for a second about the incredible reality that just two types of microscopic molecules dictate the existence of literally all life on Earth. I mean, everything from the smallest invisible bacteria to the giant oak tree outside your window, and yes, even you and me, were all built and maintained by the dynamic interplay of just two molecular giants, DNA and RNA.
It really leads us to ask this super compelling question. How does a simple sequence of chemical letters actually build a complex, breathing human body? How do a few basic molecules just sitting there in the dark of your cells know exactly how to construct a functioning heart or determine the exact pigment of your eyes? Honestly, it sounds like absolute magic. But, as we're going to see today, it's just incredibly elegant chemistry. It's simple codes seamlessly translating into complex living organisms.
So let's take a look at our two main characters. We have deoxyribonucleic acid, that's DNA, famously known for its beautifully twisted double helix ladder shape. And then we have ribonucleic acid, RNA, which kind of looks like someone took that DNA ladder and sliced it right down the middle to make a single strand. We're going to explore exactly why these structural differences exist, but I want you to keep this foundational visual in your head as we dive deeper into their specific chemical breakdowns.
Here is our quick roadmap for today's explainer. First, blueprints and builders. Second, comparing the sugars. Third, the nitrogenous base swap. Fourth, double versus single strands. And finally, number 5, roles in the cell. All right, kicking things off with section 1, blueprints and builders. Before we get completely lost in the chemistry weeds, we really need to understand their overarching dynamic. The best way to think of DNA is like a biological flash drive. It encodes all of your genetic information. It is the ultimate master storage device meant to last an entire lifetime, and it's designed to be passed down securely between generations. But, a flash drive sitting on a desk doesn't actually do anything on its own, right? It needs to be read. And that is exactly where RNA steps in. RNA operates as the active printer, or the reader. It decodes the information locked on that flash drive, converting it into a format that the cell can actively use to build proteins. So one is the highly secure lockdown archive, and the other is the bustling active factory floor.
Moving right along to section 2, comparing the sugars.
You might not know this, but the very names DNA and RNA actually come from the specific sugar molecules that form the backbone of their structures. Ribose, which is the sugar in RNA, features 2 hydroxyl groups at its base. That's just an oxygen and a hydrogen attached together, usually written as OH. Now, deoxyribose, the sugar in DNA, well, the prefix deoxy literally means minus oxygen. It's actually missing one of those exact oxygen atoms, leaving behind just a lone single hydrogen atom. Now, I know what you're thinking. It seems like a tiny, almost completely insignificant chemical tweak, right? But believe me, its impact is absolutely massive. So the crucial point to remember here is this classic rule: form follows function. That missing oxygen in DNA is completely, 100% intentional. Because DNA lacks that extra oxygen-containing hydroxyl group, it is far less reactive and much, much more chemically stable. It's incredibly resistant to degradation and harsh alkaline conditions. RNA's extra hydroxyl group, on the flip side, makes it highly susceptible to being broken down easily, which, if you think about it, makes perfect sense. DNA needs to be kept incredibly safe for your entire life. RNA is temporary. It needs to swoop in, do its job, and then quickly get out of the way before your cells accidentally overproduce a specific protein. Which brings us nicely to section 3, the nitrogenous base swap. Now let's talk about the literal code they use. Both DNA and RNA use a 4-letter alphabet to write their instructions, but their alphabets aren't completely identical. DNA uses adenine, thymine, cytosine, and guanine, which scientists usually just shorten to A, T, C, and G. Now, RNA shares 3 of those exact same letters, adenine, cytosine, and guanine, but it makes one absolutely critical swap. Instead of using thymine, RNA uses a base called uracil. So an RNA strand actually reads A, U, C, and G.
So what exactly is uracil anyway? Well, chemically speaking, uracil is essentially just the demethylated form of thymine. That means thymine has an extra little chemical tag attached to it called a methyl group. And let me tell you, that extra methyl group in thymine is absolutely brilliant. It makes DNA significantly more resistant to oxidative damage.
And even cooler, that methyl group can be modified by cellular enzymes to turn specific genes on or off. It's a process called DNA methylation, and it's a massive, fundamental part of epigenetics. Uracil totally lacks this specific regulatory potential. Now, here is the really fascinating evolutionary why behind all of this. Why doesn't DNA just use uracil to begin with? Well, it turns out that cytosine, the C base, has a really bad habit of spontaneously breaking down and mutating into uracil. So, imagine if DNA naturally used uracil as one of its normal letters. The cell's repair machinery wouldn't have a clue if a uracil was actually supposed to be there or if it was just a broken, mutated cytosine. It would be chaos. By exclusively using thymine, DNA makes it incredibly easy for the cell. If the repair enzymes are scanning along and ever see a uracil in a DNA strand, they instantly know, nope, that's a mistake, pluck it out, and fix it. Using thymine literally prevents massive, unavoidable genetic errors. Okay, let's jump into section 4, double versus single strands. We've spent a lot of time on the microscopic chemistry, so let's zoom out a bit to look at the incredible physical contrasts between these two. As we know, DNA is double-stranded to make it incredibly stable. But it is also massive. If you took a single human chromosome and completely unraveled it, it would be up to 2.3 meters long. To fit inside a microscopic cell, it has to be tightly coiled and kept securely tucked away inside the nucleus, kind of like a highly protected VIP reference library. RNA, however, is almost always single-stranded. And because it only copies very small specific fractions of the DNA sequence at a time, just one recipe, not the whole cookbook, it is vastly shorter, maybe just a few thousand bases long. This super small size is what allows RNA to easily leave the nucleus and move freely out to the cellular factories. And finally, that leads us to section 5, roles in the cell.
So how does this whole structural showdown actually result in building a human body? Functionally, here's how it breaks down. DNA replicates, securely stores the master blueprint, remains highly stable, and never ever leaves the safety of the nucleus. RNA, acting as that crucial intermediary, transcribes the code, translates it into physical proteins, is highly temporary so it doesn't clutter up the cell, and does all of its active work out in the cytoplasm.
And here's a fun fact. RNA doesn't actually do this massive job alone. It splits into 3 highly specialized forms to get the work done. First up, we have messenger RNA, or mRNA. This acts as the cellular courier, copying a specific gene's code from the DNA and safely carrying it out of the nucleus. Second, we have transfer RNA, or TNA. These little guys read the messenger's code and fly around fetching the exact amino acids required for the recipe. And finally, we have ribosomal RNA, or rRNA, which actually forms the factory itself, the ribosome, where those amino acids are physically stitched together into a completed protein. Now, this is where things get truly mind-blowing regarding that last step. Ribosomes are actually ribozymes. What does that mean? Well, for the longest time, scientists firmly believed that only proteins could act as enzymes, the active catalysts that drive chemical reactions in biology. But it turns out the core mechanism inside the ribosome that stitches those amino acids together isn't a protein at all. It's the ribosomal RNA itself doing the heavy lifting. RNA isn't just a passive messenger dropping off a blueprint, it is actively catalyzing the assembly of life.
Which leads us to an incredibly profound place to wrap up today's explainer. Because RNA can both store genetic data, just like DNA, and actively catalyze chemical reactions, just like a protein, many scientists today subscribe to the RNA world hypothesis. The whole idea is that billions of years ago, long before the complex dual system of DNA and proteins ever evolved, early life on Earth might have been driven entirely by RNA. It was the original blueprint and the original builder all rolled into one. I want to leave you pondering that incredibly powerful thought, the very real possibility that the simple, single-stranded messenger working inside your cells right now is actually the ancient ancestor of all life. Keep asking questions, keep learning, and I'll catch you in the next explainer.